Osteogenesis imperfecta, also referred to as OI or brittle bone disease, is a genetic disorder that causes bones to break easily. These breaks can occur from little trauma or from no evident cause. There are four types of osteogenesis imperfecta. Type 1 is the mildest form of OI. It presents itself in childhood with fractures often caused by mild trauma. The number of fractures decreases in adulthood. Type 2 osteogenesis imperfecta is the most severe form. Type 2 impacts infants even before birth. These children have bones that appear curved or crumpled and are often fractured before birth. They have fractured and deformed ribs, underdeveloped lungs, and a narrow chest. Infants with type 2 OI are often stillborn or die shortly after birth because of breathing failure. Type 3 osteogenesis imperfecta is also quite severe. OI type 3 causes infants to have soft and delicate bones that often fracture before birth or in early infancy. Due to rib fractures, some infants have life-threatening breathing problems. Malformations of the bone often worsen over time and impede the ability to walk. Type 4 OI varies the most of all forms of OI. The symptoms are able to range from mild to severe. According to the National Human Genome Research Institute, about 25% of infants with OI type 4 are born with fractures. More than 90% of osteogenesis imperfecta is caused by a mutation in the collagen 1A1 and collagen 1A2 genes. These genes provide instructions for making proteins that are used to assemble type 1 collagen. Type 1 collagen is the main protein in bone, skin, and other connective tissues that provide strength and structure to the body. The majority of osteogenesis imperfecta mutations occur in the collagen 1A1 gene, causing type 1 OI. These gene mutations cause the amount of type 1 collagen produced in the body to lessen. The more severe types of osteogenesis imperfecta, such as types 2, 3, and 4, occur because of mutations in either the collagen 1A1 or collagen 1A2 gene. Mutations of these genes usually change the structure of type 1 collagen. The majority of osteogenesis imperfecta disorders are inherited in an autosomal dominant pattern. This means only one copy of the mutated gene in each cell is enough to cause OI. Most people with type 1 or type 4 osteogenesis imperfecta inherit OI from a parent who has the disorder. In cases where a child has the dominant form of osteogenesis imperfecta, but neither parent has OI, the disorder is a spontaneous mutation. More rarely, osteogenesis imperfecta can be inherited in an autosomal recessive pattern. This means in each cell, two copies of the gene must be altered. In these cases, the parents do not have osteogenesis imperfecta, but they each carry one copy of the mutated gene. In order for the child to have the disorder, the child must receive one copy from each parent. According to the National Human Genome Research Institute, osteogenesis imperfecta occurs in approximately 1 in 20,000 individuals, including people diagnosed after birth. Also, OI occurs equally among gender and ethnicity or racial groups. A family history of osteogenesis imperfecta is one way to diagnose the disorder. Doctors may also look for indicators including frequent fractures, short stature, a blue tint to the white part of the eye, teeth problems, and hearing loss that progresses after puberty. When these indicators are present, doctors might order a few different tests. X-rays are used to see fractures at different stages of healing and an irregular skull bone pattern called Warmian bone. Lab tests can also be done. DNA sequencing of collagen 1A1 and collagen 1A2 can be done to identify the type 1 collagen gene mutation. Also, a small skin biopsy can be done to detect changes in type 1 collagen indicating osteogenesis imperfecta. Osteogenesis imperfecta has no cure. Treatment is determined based on the severity of a person's symptoms. The positive treatment outcomes are to control the symptoms, maximize an individual's mobility, and develop optimal bone mass and muscle strength. Fracture care, substantial surgical procedures, and physical therapy are often suggested for people with osteogenesis imperfecta. Wheelchairs, braces, and other mobility aids are commonly used, especially in more severe cases of OI. The life expectancy for someone with osteogenesis imperfecta depends on the severity of their disorder. Infants with type 2 are often stillborn or die shortly after birth, while people with type 1 can live to the average age. Recently, a new medication treatment has been explored for treating osteogenesis imperfecta. Bisphosphonates are a class of drugs that prevent the loss of bone density. This medication is being used to help with bone formation and to decrease the need for surgery.